Okay, this is the second half of Chapter 15 on poliovirus. We're going to go through the poliovirus replication cycle. Thankfully, it's a pretty simple, straightforward cycle. And then talk just briefly about some of the eradication efforts. Okay, so poliovirus is non-enveloped. It is a icosahedral virus, and it has a positive sense single-stranded RNA genome that's quite small, less than 10,000 nucleotides. And as we've talked about in the past, right, this genome can basically act as messenger RNA, be translated directly into proteins, so there's no need for the virus to enter the nucleus, which, as I said, keeps things kind of simple. As we've seen with other viruses with this genome type, you have a built-in, 3' poly A tail, built-in 5' secondary structure with the IRS that's going to act as that 5' cap. And in this case, you have the VPG, which is the virion protein, genome-linked virion protein there, and that's going to serve as a primer for genome replication. And also, as we've seen previously with other viruses in this kind of setup, you're generally going to get a polyprotein that's translated. Then that's going to be cleaved by proteases into individual protein components. And you'll get 11 mature proteins out of this, as you can see in the diagram on the right here, loosely grouped into the capsid proteins, VP1 through 4, and then everything else, the non-structural proteins, they're calling it. Of note, there you've got 3D Pol, which is the polymerase. And here's just the full list of proteins. You may notice if you're counting, there's more than 11 here, and that's because you have some combos. So for example, you've got 2A, 2B, 2C, but then you have 2BC, or similarly down here, you've got 3A, protein 3B, and then 3AB. So with some of those combos, you end up with more than 11, but same basic idea holds. So let's step through at least a few parts of the replication cycle in terms of attachment and just capsid structure. So I know we talked in one of the earlier chapters about icosahedral detailed structure, and you have 4 capsid proteins, so VP1 through 4. 1, 2, and 3 are going to form the triangles that are going to make up your icosahedron, the equilateral triangles. VP4, you don't see it here, but it's associated with the inner surface of the capsid wall there. And the VP1s, where they come together here at a vertex, you'll notice you get in light yellow what kind of looks like a star there. They call this a plateau or a mesa. You can see it in the more detailed image on the right with some 3D relief there. So you get this kind of star-shaped plateau, and then you get like a deep trench or canyon around it, and that's where the cell surface receptor protein is actually going to bind in that canyon there. And we've mentioned this protein before, but the cell surface receptor for all 3 poliovirus subtypes is CD155, aka the poliovirus receptor. And I brought this up in an earlier chapter talking about host range, where some researchers had done an experiment showing that if you express this protein in mice, which normally do not have a poliovirus receptor, then they become susceptible to poliovirus and get the paralytic polio symptoms and all that stuff. So it just shows how important this step can be in establishing the infection. So this protein is a glycoprotein. I think it's normally part of adherens junctions, and you find it expressed on epithelial cells in the intestine, as well as M cells that are part of those Peyer's patches that we mentioned in the first video. So here you can see that poliovirus receptor, right, docking into the canyon, as mentioned before. They think it's getting taken in by non-clathrin, non-caveolin mediated endocytosis, and there's going to be some conformational change that's happening to the capsid inside the endosome. And then the VP1 protein, so let me go back, this guy, right, is going to insert into the endosomal membrane, creating a pore, and then the viral genome is going to get out into the cytoplasm, as you see here, with that VPG attached at the 5' end. And we're basically immediately going to get translation and the cleavage, as we discussed. And once the 3D Pol, the polymerase protein, is produced, then we can make a negative sense anti-genome copy and then crank out some more positive sense copies of the genome using that as a template. And we don't get a whole lot of detail on this, but apparently a lot of this replication activity is happening in little membrane-bound vesicles in the cytoplasm. And we have seen, similar theme with other viruses where we've had some ER remodeling and things like that. I'm not sure exactly what the source of the membrane is in this particular case, but we have seen that idea before. So then we've basically got everything that we need to create new virions, and they have a fair amount of detail on how assembly happens. So you'll get VP1, VP3, VP0, all form little structural units that are called protomers. 5 of those protomers will group together to make a pentamer, and then you get 12 pentamers grouping together to give you the full, what they call the procapsid, before it's fully finished and closed off. Then you'll have your positive sense copies of the genome, right, associated with VPG, capped in there, packaged in there, and everything closed up. VP0, so maturation is going to happen when that VP0 protein gets basically chopped in half, one half becoming VP2, the other half becoming VP4, and then it's simply going to get out either by lysing the host cell open, or it seems to also be capable of co-opting some of the vesicles that may be involved with autophagy, so sort of that lysosomic vesicle trafficking pathway. So, as I said, not too bad in terms of replication cycle there. And it's definitely worth talking for a few minutes about some of the eradication efforts, because this has been really a heroic effort to try to eliminate polio. We haven't quite gotten there, but we're pretty darn close. As we'll see, this was initially a project that was started in the late 80s, and the initial goal was to try to eradicate polio by the year 2000, calling it a gift from the 20th century to the 21st century. We did not hit that mark, but again, we did get pretty close. So this was a huge effort, right, involving, governments, all kinds of government organizations, all kinds of privately funded organizations, charitable organizations. So there have been a lot of players involved with this effort. And I want to just talk about what are some of the things about polio that make it either ideal or not so ideal target for eradication. We'll start with the good. So poliovirus, as I mentioned previously, only infects humans, so you don't have to worry about animal reservoirs. That's a plus. We have only 3 serotypes existing, so it's a relatively small number. We're not usually getting persistent infections, and we know the polio vaccine can confer lifelong immunity, and in particular the oral polio vaccine, very easy to distribute and administer since it can be given orally. But despite those positives, you do have a few negatives. One is, as we talked about in the first half of the chapter, 70+% of infections are asymptomatic, which practically speaking means you can't just do like targeted ring vaccination. You really have to vaccinate all kids, or a very high percentage of children, to effectively stop the spread of the virus. And another downside is that you need multiple doses. They estimate typically 4 doses to get full immunity. So if you're talking about vaccinating every child on planet Earth 4 times, right, that's a huge logistic hurdle to overcome. And I mentioned this previously as well, but that oral polio vaccine from Sabin, right, was selected because of how easy it was to use, also cheaper to produce. But as I think I alluded to, one potential problem there is because it's live attenuated vaccine, it could revert at some low frequency to the wild type and actually cause disease. So, of course, if we have to immunize all children, really have to focus on high coverage, right? Trying to hit as many kids as possible. So this meant, surveillance efforts, just going door to door, trying to find as many kids as possible. And a lot of this was, as I said, very heroic, because you had, some of these countries are not the most stable geopolitically speaking, so being war zones, right? People risking their lives. They're very hard to get to locations in some cases. So you had people really going out of their way and potentially risking their lives just to deliver vaccines to children. But that being said, they did a great job. So just to give you an example, in the mid-90s, they vaccinated over 93 million kids in a single day in India. So that's incredible. And just a little timeline here, as I mentioned, late 80s was when this initiative was announced. At that time, there was about 350,000 cases of polio documented, at least worldwide. And then just less than 10 years later, we had dramatically reduced that number, so we're under 4,000 cases reported. By 99, we had at least one of the 3 serotypes was no longer in circulation. And by 2006, you only had 4 countries, so India, Nigeria, Pakistan, Afghanistan were the only 4 countries where wild poliovirus was evident, although you did have some of the vaccine-derived polio in some other countries as well. So if you want to see the latest, I'd encourage you go check out this link, Polio Today, polioeradication.org. It'll show you where all the documented cases are as of 2024. But we're getting there. So we are down to just a handful of countries and a small number of cases. So that's encouraging. Although as you're aware, there has been some resistance to vaccines in general, not just in the United States, but I think that's been worldwide. That has kind of made it more challenging. And of course, the fact that you have some of the vaccine-derived polio that's actually circulating now and being passed person to person. So that has slowed efforts as well. But I think it's still possible. I think we can get there. It's just a matter of do we have the will to get it done, and if so, I think it can be done. So that would be a huge achievement for humanity. All right, I will stop there.